This is my layman's terms.
I was like, yes, nip it at the bud, root it out.
Yes.
He's like, don't eat them paint chips.
Both are accurate. I'm going to go at the root. So now you can take skin cells, turn them into stem cells, like the original stem cell, like the earliest stem cell, then tell that to become anything else in the body because it has unlimited potential. It's immortal. It can just live forever, divide into anything. It's immortal. That is the hallmark. But yeah, I'm in the lab. I got my lab coat. I'm got the gloves on.
I'm glad you have a lab coat. That's important. As a doctor, you should definitely have a lab coat.
I think there's like a study that showed that people like just trust people with lab coats.
100%.
Like even if they're not actually doctors or anything.
Blindly. Don't even ask a question. Absolutely. Do my gynecological exam. That's fine.
Get what you were talking about, but like
tangent.
It's creative genius, okay?
But you know, it might be relevant.
Hi folks, welcome to the pod. Hope you're beating the summer heat, or maybe you're listening to this in winter and we can live a little vicariously through you to escape this dredge of summer. Welcome to the hot, steamy, newest episode of We're Experts Now. I'm your co-host, Rebecca.
I am your other co-host, Jackie, and I don't know if this episode's going to be a hot and steamy one, but it will be fascinating. So we'll get to that in quite a moment. Also, I think when this comes out, we'll be just on the edges of fall.
Oh.
And so we're almost there, Bex. Like we are actually escaping the summer. We're getting into our cozies here in a couple weeks. We have some exciting things coming in October. So listeners,
oh, that's right.
beware. Very exciting stuff. But
some spooky plans for this episode, for this pod.
The spookiest, some might say. Actually, I don't even like horror films, so no thank you. What have you been up to?
Oh, you know, I've been out here doing amateur things as I always am. I'm owning a car for the first time that is not, it's like one step above a lemon. It's a lime, I guess, you know, like it's kind of zesty, but it's still a little testy. And it started leaking inside my car, which I was like, I didn't know you could have an inside a car leak.
Was it wet recently?
It was wet, very wet. Well, no, not outside in nature, just on the floor of my passenger side of my car. And It was very embarrassing, you know, trying to go on dates, pick up my date and be like, yeah, there's a bucket down there. Don't mind that. It's fine. Just, you know, trying to save on water this drought season. But I, through some Googling, found out that I had a clogged evaporator drain. So just because the water is so crusted with minerals and grossness that it just built up in my car. And basically I had to have someone come out and just like pop a hole in a balloon. You know, it was like something I could have done if I had the right tool and the know-how, but I went with my mobile mechanic guy. And it's also embarrassing because he's a mobile mechanic. So he came to my nice neighborhood where my junk car that always gets the We Buy Junk Cars business card stuck in it is just sitting there, and he's under there, and he gets I think it was basically the air pump that you can like the portable air pump that you can use to fill your tires. He just took that and then like put it in the clog, and then all this water just rushes out onto the street. And I'm like, sorry neighbors, I hope, you know, it's just water, it's not a chemical. I hope it's just dirty water, good for the ground. It clogged, flushed me out,
not good stuff, and you're just killing all the bugs. I mean, I don't know what, and there are no animals in LA, so actually you're fine. You're not killing anything.
And yeah, that was it. It was a night, and I was glad it was like not something cracked, you know, because then I'm like, I'm gonna have to get something replaced, gonna buy parts. It was actually only $40, so that was worth it. And now I learned a little bit more about my car every time something goes wrong with it. And yeah, nothing long term. Just got to flush out some toxins, and now my car just, you know, smells a little bit like mildew. But that's the next problem we have to tackle.
Well, we love that, but. No, we don't. Actually, no, I don't love that. But we should definitely have a mechanic on the show sometime, because I feel like that's something I have tried to learn to do, change my own oil. And once I realized that I wasn't really even strong enough to open the little oil container compartment, I was like, no, I'm done. It's hot and I'm hungover. So
Yeah, and it's like then you have to invest in having all the tools and a space to do it, and it's
yeah,
the time and effort. Sometimes it is nice to just like go get it taken care of, but it's also, it would be great to have like, yeah, like
the
if your
knowledge.
dad didn't show you how to do this, this is how you change a tire if you're
Okay.
stuck on a
Well,
freeway.
don't bring up some sore subjects, okay? I
Hey, mine
had to
didn't
teach
me
myself.
either.
I had to teach. Well, so Carl, who was on a previous episode for boot camp, once made fun of me because I said, like, I was like, I don't know how to do this thing with my car, and he's like, Jackie, your dad died a long time ago. You got to stop using this excuse. But I can change my own tire. I can
Okay.
top off my oil. I
Yep.
can check all of my stuff. Like, I know the basics. And I think it's important to know, like, if there was like no other option and I had to change my oil, I could do it. At this point, I now have the knowledge. It would take me 5 hours, but I could do it. So it's important.
You're ready for the apocalypse. Yeah, we need someone to just come on and teach us the basics, and then also like how not to get swindled when you go to the mechanic, and how to properly not get swindled buying a new car or something like that, you know?
So the trick is actually know a mechanic. Ryan, who
True.
did astrophotography on another previous episode, he is a mechanic, and I often call him and I'm like, Ryan, is this a real thing? Does it seem like this? I would possibly do that to my car? Okay, yeah, okay, I'll pay for it.
It's all about who you know. And what are we getting to know this week?
Okay, girl, I got a
Uh-oh.
doozy for you. It's going to be one of the ones that we absolutely love learning, and like the facts are going to be so mind-blowing, but we're going to know nothing about the subject. So we're going to get into the world of drug discovery. In particular, I don't mean that you and I are taking drugs and discovering.
I might have discovered 1 or 2 drugs in my time.
No, that's not the what we're doing in this episode. This episode, we're going to talk to a friend of ours. I'm going to do the best I can explain of what she does so we can get into kind of how we think that works. But she works particularly in neurology, or neuroscience, I guess, looking for the pharmaceutical drugs and how they react to your brain. And I think they probably treating things like MS and those types of neurological, maybe Alzheimer's, maybe Parkinson's. Like, I actually don't know.
A lot of maybes already.
I did ask her some questions because I was just so fascinated when I found out this is what she did. So I do have a little bit of knowledge, but I got it 2 months ago and or a month and a half ago, and it was Pride weekend, so I was definitely had a couple drinks. But yeah, so that's really where we're going to start, is how you take chemicals or components. I think they use some cells as well, like, right? So like, I don't know, like maybe this venom that we found in the Amazon, mix that with some stem cells and then make something like a drug, and then you test that on stuff.
Yeah, I would imagine. I mean, it sounds like we're dealing with a smarty pants. This is someone with a science background. And I think there's things you learn along the way in your science journey that kind of prepare you for this kind of research, where you're not always just starting from scratch. You're sort of using past studies and discovery to be like, okay, well, this compound is this kind of structure, so it's probably going to react this way. We're solving for this kind of thing. But yeah, I think, it's always trial and error and new discoveries all the time. And I mean, there's certain things like prions I know are really hard to, like mad cow disease. It's like really hard to solve for things like that. Like, we don't know or certain viruses that are like technically incurable. And
well, I think that's what I wanted to know. Like, this is one of the questions that I asked her. I was like, are you trying to, for example, multiple sclerosis? You know that you want to solve for multiple sclerosis. So I think in that scenario, the company would start with the knowledge that they have on multiple sclerosis, what's worked, what hasn't worked. How do we combine those knowledge and, create a new drug, or how do we go down or improve off of something that's already been done? So I think there's that piece. And now what I had asked her, and because I thought it was really important for us to know, what she is doing is they don't actually have something that they're solving for. They're just seeing what happens, which I also think is really cool, because I think I read not that long ago an article talking about like the illnesses that aren't very common. Pharmaceutical companies at times don't pursue going after them as much because there's not as many people with it, right? They're trying to solve for the illnesses that have the most people, have the most impact, right? So I think that this is really interesting because they could solve for anything, right? Because they're not trying to pick one area like, oh, I'm trying to, solve for I don't know, cancer or whatever it may be. They're just saying we know we want to work with brain stuff. We know we want to work like in neurology and then neuroscience, and we want to see what happens when we add X, Y, and Z together. And so I think this is a good place for us to start of how do you add X, Y, and Z together? Because that, I was like, you got to have a lot of petri dishes, right? Well, and yeah, right. And then you usually need to keep things like cold or hot depending on what you're working with. Like, so I imagine that you need to like cryo freeze some specimens that you like, maybe you keep them in an incubator for a while because they can grow. Usually heat grows stuff, cells and molds and funguses.
And that's how we got this recent outbreak of ancient rubella and measles, because some of that broke out of the lab. It's like you gotta keep that stuff
frozen.
Yeah.
Keep it on ice. And then I think some stuff you're heating, right? So like, I think you have a lot of equipment to store all the stuff you're working with. Like I read an article recently that it was like a company, a lab, or like a college, maybe it was suing the janitorial services because the janitor had accidentally unplugged something, even though there was like a note on the wall that said like, don't turn down this switch, don't unplug this. It was like all over. It said, don't do this. The janitor did it, and it destroyed that person's 20 years of work.
Oh my gosh.
Because they had been like incubating something for like 20 years. And like, cutting edge science. So I think there's a lot of not just creation and testing, but there's probably a lot of equipment to store things properly.
That's like when you don't press save on the paper you've been working on and just...
No, Bex, that's not the same. That is like totally different circumstance. And one is a rubella outbreak and one is Rebecca has to spend 4 more hours writing that paper.
Okay, yeah, it's a little different, but I had no more drug jokes. We're dealing with a professional here, but also it's like that sounds like a lot of shooting in the dark. It's like what then are they even starting with? Are they starting with drugs that already exist or new compounds and how they can be used? Like,
well, I think that's it. All drugs are a compound, right? So you're probably totally hitting it right there. They're starting with drugs or compounds and parts of compounds that have worked previously. And I imagine that they're taking all that science and adding it together and being like, okay, and study XYZ, it said that, this compound really worked, and study ABC, it said this other half of this compound worked. What happens when I put them together? Or what happens when I put them together and I make them hot? Right? Or I make them cold, or I slow it down, or I add another chemical compound to it. And I imagine that's what they do. So like, I feel like I'm going to use a kitchen as an example. Like, right? Like I'm making a different dish each day, and I'm then like checking to see what happens with that dish over time. Because sometimes if you let a dish sit overnight, it's better the next day, right? Like tacos are often better the next day. I'm going to get some hate mail on that one.
Yeah. Cold pizza. It's the best. And well, because I know that's how we got Viagra. It was supposed to be a medication for something else, I think. I can't remember what it was supposed to be for.
Well, and that's Botox as well. So Botox
botulism
came from migraines. So they were using, so I actually get Botox for migraines. I get, for the listeners, because I'm sure people are really going to want to know this, I get it in my eyebrow line and then my head, like hairline, like you can't really see it. It's like right here. And then I get it down my neck and into my shoulders. And they were testing for migraines because essentially it freezes your muscle, right? And so they have a theory that a lot of the muscles back here tensing up is what can give you migraines. And so they freeze those muscles. But when they started testing on that, they realized, oh, wow, people's faces are freezing. Some people might like this. And I've had my shots done by 5, 6 different people now. Each one does it differently. So I've had one that did all in my hair and not in my neck. I've had ones that have like done mostly in my hairline up here and none in my eyebrows. So like there's different like locations that they all go into. So I think that like I definitely like, oh my God, I'm gonna regret doing this, but I can't really move my forehead really that well. Like it's not like, so, and that's how they figured out that they could sell it for, right? Like, so Botox is a perfect example of that. And Viagra, a bunch of different things. Like, I think Viagra is a heart medication.
That's what I was thinking, but then I was like, doesn't it give you heart problems if you take too much of it?
I feel like it probably either thins out your blood or like decreases or increases your blood flow. Right? Because like you want blood to flow to the peen.
To the peen.
So, to the peen. We are children. But on this episode, we're talking about neuroscience. So my Botox reference was actually way more helpful. But yeah, I think you take stuff that you already know. I know that she works with stem cells. I know stem cells are really cool. And that there's a lot of
very divisive.
Yeah, you could do so much with it, and there's so much. I think because it creates growth, right? Like it stimulates growth really quickly, and I imagine that's helpful because you want to like you don't want to wait 6 months for a tumor to grow in a lab to see how like something affects it, right? Like I imagine you want things to grow really, really quickly, and I don't know if stem cells help with that.
I know they're very hotly debated because of the conservative views on life and conception.
I am very pro stem cells. I'm going to say it here. I'm going to say it now. I'm going to say it proud. Like,
but it's interesting that there are those crossovers where it's like you cross over into moral dilemmas too. Like what is the moral implication of some of these studies? And then I imagine, there's like maybe testing and you're in a lab and then at some point you wanted to go to trial, right? You want to like see how it works. And there's only so much you can do in a lab, as Oppenheimer said.
Well, yeah. So you have your clinical trial phase. So you have your discovery phase, then you have your clinical trial phase, then you have your kind of go to market strategy, your like medical, awareness, like, you have to then train people on a drugs. So I think there's the process of getting a drug to market is very, very long. And I think they very much work in this very narrow, very part. I'm using a lot of words right now. This very narrow part of the industry of that like discovery phase. And I think that's probably mostly in a lab. I'm going to, again, as usual, know the business side better of things.
I know you said go to market and I was like, I feel like that's not science. It
is not science, but that is part of the process. I don't think that her company or she works in that. Like, I think she works just in the drug discovery phase. I think my understanding of the pharmaceutical industry, there's a lot of startups, right, that do drug discovery, and then a lot of them get bought up by the bigger pharmaceutical companies. They get a big buyout because they've discovered something, and now that company wants to like introduce it into the rest of their like drug portfolio, right? It might complement another treatment that they have. So say if she works in neurology and she discovers a great drug for MS, then I imagine that a company that already has drugs that help treat MS is like a big one, is probably going to want to buy up that company really, really quick.
Yeah.
So I think these startups are the companies that are a lot of the time creating that information and like really doing that discovery that probably a lot of the bigger companies aren't doing, because I imagine a lot of them stick to their portfolios.
Yeah, and I'm going to be curious too about whether that information stays private or if it becomes open source, because I know sometimes people are more invested in, like, we just want the cure. We want people to know about this. I'm going to make this information available to other scientists versus this is owned by, X company and it's proprietary information and it's not part of the free scientific world and community. So, yeah, I wonder, I mean, obviously you don't probably as a scientist have too much say in how that happens. Just, you pick the lab you want to work for and where you think your skills will be best put to use and go with the flow.
I'm also interested to find out, like, what is tracked and why is it those items that they are tracking, right? So, like, say that you're trying to find this treatment for MS and you get a lot of fails. Like, what do you do with that data? Like, is that data shared outwardly, right? Because do you want to stop a lab in, like, India from doing the same test that you just did a second time and wasting their time? Or is, like, only the success shared, right? Like, do you only really tell people, oh, I found the answer, not that here's a lot of data? Or is that only shared if your company gets acquired or something like that? Because I feel like there's so much information that comes along with an answer, right? Like, what happens with all that other information?
I also, like, I'm picturing this process as like, okay, I've gone to school, I'm a scientist, I've gotten my job, I've gotten maybe some papers published or, co-authored some papers to kind of boost my resume. Now I've gotten my job, now I'm in the lab, now I know what I'm going to be working on. Is the application mostly writing equations on chalkboards and papers and kind of figuring it out in theory? And how much of it is actually like, here's the chemicals and the droppers and putting things together? And then also, like, how do you measure the output? Because it's like, I can look at a paper equation and see what the output is supposed to be, but it's like, how do you actually measure these, like, very fine, small molecules and atoms of differences, and like what kind of devices you have to use to test, like, the output of certain chemical reactions? It's like, is it looking at it under a microscope and seeing the atomic structure? Is it like, is there a chlorine testing
Probably a
strip?
lot of testing. I think that, like, so a couple things. I think that whoever is leading the company or the department or that project is probably the one doing that big picture. This is what we're going, this is direction, we're going to use these drugs or these data sets to, like, build our known knowledge off of before we grow out. And then I think a lot of people probably under them are, like, doing the testing, right? So, like, I go into the lab every day. I either, on Mondays, I create stuff. So I mix these 6 things together, or I mix different things together different days at different temperatures at different times. And then I test it over the week and or 6 months or however that testing period looks. And I think you hit it, though. I think depending on what you're doing, like, they probably have cells, right? So they probably have human cells that have that illness or have that whatever you're trying to fix, right? So if it's neurological, a lot of the times I think it's like when your synapses are not connecting. So, like, why are those synapses not connecting? Why is that one part of your brain not lighting up or whatever that may be? How do the cells in that area respond to what I just mixed together? And then I think it depends. Like, you probably look at it under a microscope. You probably test its chemical compounds. You probably, look at, like, see what's happening. Like, is there a smell? Is there, you probably test a whole bunch of stuff every day. And if there's any change, you probably track that change. And then once you probably start to see trends, I imagine you dive into that. And that's what the lead of the project would do. And
I like picturing a scientist, like, dipping a finger in and then tasting it, like, yep,
I don't
that's
think
arsenic.
They taste it.
Oh, that's arsenic,
I don't think they taste it. But they, like,
smell test, wafting.
Yeah, I think that they push in directions, like, right? Like, oh, we want to see, for example, like, we want to see regrowth in this area. Like, oh, the cells regrow when we add this and, like, this and under these temperatures. What happens if we added a degree to it now? What happens if we add humidity to it?
Give it a bachelor's degree, get it some education.
But, like, or, like, like humidity, what happens if we add all these, like, what if I add another thing that I know makes growth? So I think once you start getting answers, you deep dive into them and try to understand the why. why
Also,
did this change?
do they use Kelvin? Do they use metric? Like, what's the... I feel like
I
scientists are
imagine with
better at metric.
drug discovery, you're working with the human body, so I imagine most of the time you're probably not using Kelvin because that's really hot.
Oh, yeah. I don't know what
But like
Kelvin
they
is.
could be using Celsius. I don't know if they use Celsius or Fahrenheit. I imagine they probably use Fahrenheit because we're in America and we are like that.
I feel like scientists are using Celsius.
I feel like
They're
I would
using
believe that everywhere
metric.
except for the US. I bet they can do it in both pretty easily, but I don't know what the science community uses. I do know that the science community often is based off of French. A lot of it's in French. It used to be the universal language of the medical community.
Oh, well, that makes sense because I feel like there's a lot of French scientists that like cured Pasteur, Louis Pasteur,
Love you naming people. Okay.
Mendel. Not French.
What else? This is a hard one.
I'm ready to get in the lab. I think let's go be some lab rats.
I'm not gonna lab rat. I will hang out in the lab and watch you be a lab rat.
Guinea pig time. Let's go. Welcome back, intrepid explorers. It's time to put those lab coats on, your goggles, your required safety gear, and put our hypotheses to the test. Jackie, who do we have in the lab with us today?
Well, we'll be asking a lot of questions, a lot of who, what, when, where, why. I know that's the news, but that's really where I know how to start. We have a new friend of mine, but she lives locally, so we plan on hanging out many, many times from now. But Devi, I tried to explain what you did in part one, so I'm going to let you actually do that because I know you're going to be far more better at it. But tell us a little bit about yourself and your background.
Yeah, sure. Also, I have to say, you guys did a great job in the part one.
Hey.
Yeah, I was actually very impressed. So yeah, thank you for having me on here. I'm very excited to talk science and drug discovery. So a little bit about me. I have a PhD in genetics from the University of Wisconsin-Madison. And so I worked in a neuroscience lab.
She's a doctor.
I'm a doctor.
Dr.
Yes.
Devi.
Yes.
She's a doctor. Okay,
but like the non-medical doctor, like don't ask me to like, diagnose you. I can't. But I could talk to you about weird science things. Yes, happy to do that. So my degree is in genetics, but my research was in neuroscience. So I was basically studying how the blood vessels in your brain develop. So all my like hands-on research in the lab was with mice and with like the brain. And so then after I graduated, I joined a lab, a small company in California, and we worked in drug development, drug discovery, but mostly focused on like cancer. And then 2 years ago, I started at my current job called Boost Neuroscience, another little startup. But we're working on drug development for neuro-related diseases. So I think Jackie actually explained this pretty well last time in the part one, that like we are taking kind of a different approach than most companies and most people. Where we're kind of going after the biology as opposed to the disease or the symptoms. So you guys did a great job of like kind of hitting like some really great questions and some very random questions that I'm like so curious why you went there. But on one hand, I have to say I'm relatively new to drug discovery actually. So like I can give you like my experience and my like high level knowledge of what people do. And kind of like what you guys were saying in part one, I think the most common thing people do is go after the disease, right? So like you have a disease, you have like symptoms, like whether it's, cognitive decline, like you're losing your memory, or, in other fields, like, diabetes, you have like problems with insulin or pain or something. There's like very clear symptoms. And most people go after the symptoms, right? You're trying to prevent or reverse whatever is going wrong. And that's one approach, and that's what most medications are based off that. And then in the last maybe like 50 years, people, as we've like learned more about basic biology in various fields, we can understand more about what causes the diseases. And if you know the cause, targeting that is like way more efficient, because then you're not trying to like fight a battle that's already like halfway lost. You can go after the main like reason something's going wrong. So like
So like paint chips. We should not eat paint chips that have lead because that led to cancer. And if you can not have the cancer, that's winning, right?
that is winning.
Right? And that's like
Sure.
that's beating it to it, right? This is my layman's terms.
I was like, yes, nip it at the bud, root it out.
Yes.
He's like, don't eat them paint chips.
Both are accurate. I'm gonna go at the root that Rebecca was saying. But yes, basically. But yeah, like if you know somebody's eating the paint chips, obviously making them stop is
Tell
one
them
way.
to stop.
Yeah, yeah.
You can call me a doctor now.
Dr.
Okay, speaking of us being doctors, 1 to 10.
Oh,
how did we do?
So okay, I have to say it was a very interesting mixed bag. I was like re-listening to your part 1 like just before this, and it was like something I was like you were like so spot on where you were like, you know, like your examples of like Viagra and like Botox, yes, like totally things that were like repurposed, like perfect. Your comments about how like almost all of the drug development, drug discovery is based off of pre-existing knowledge that people have already identified and have some understanding of like what's happening in that situation, and then like trying to like fix it from there. Yes, like for sure. Things about temperature. I was like, well, I don't know what that was about. So anyway, so I think on average, I want to give you like a 6 out of 10 because like when you were right, you were like, you guys are really right. When you guys were, I won't say you were wrong, but you went on some like I don't get what you were talking about. But like,
let's call it a genius, okay?
But you know, it might be relevant. I was like, it's not in my field, but you know, I have neuroscience is one very specific subset of drug development. So 6 out of 10.
You don't get things hot and cold all day. You don't just like
sit
there with a
bag of ice. It's generally frowned upon to like freeze people. So like generally what we want to do is mimic what happens in the body. So like like when we're actually doing the testing of whatever, you know, drug we want to work on, things should be at like, you know, 98 degrees Fahrenheit, 37 degrees Celsius.
Okay, but love to like drugs come to you on ice.
Yeah, do they come on ice though?
They do come on ice, yes. So that's to like keep them preserved. So that was correct. Totally want to keep them.
Make a cocktail.
Keep them stable.
Put a lime on it.
Okay. Okay, we're tangenting again. All right. So, okay, how do we start? What is the principal scientific thing that we start? Like, is it the hypothesis? I feel like that's what they taught me in the second grade.
That's great. Hypothesis is always great.
Ding, ding, ding.
I love that. I think I was kind of thinking of this like drug discovery is such a broad term, and I was like, let's just check that we're on the same page. Like, what does that mean exactly? Because like it's actually a very complex process to go from like, I wonder if this thing does something in like neurons versus like, eat it, it's going to make you healthy. Like those are like the beginning and the very end, right? And the whole process can take like 10 plus years, billions of dollars. And I've only ever been the very, very beginning. I haven't done all the steps. So take everything I say.
How do we get from farm to Coachella? How
exactly.
do we get...
So at a basic level, the idea is you have either a disease, a symptom, or some sort of target that you know, like in our case, I'll tell you, we're trying to improve the synapses of neurons so that, as Jackie correctly explained in part 1, your synapses are your fundamental units that help your brain function within the neurons, within those brain cells. So if you could make those better, theoretically you could improve cognition and also prevent things like dementia, Alzheimer's, et cetera, et cetera. So I kind of think of that as disease, symptom, or biology. And generally people, like companies, will target disease or symptom because it's like with Alzheimer's, the disease is cognitive decline. And so those are the symptoms, but we don't know the biology actually. There's no like, if you were to look up like what causes Alzheimer's.
It's not that plaque. It's not that plaque we all hear
about. The plaque is there. The plaque is also a symptom. Yes, the plaque is definitely like there in people that have Alzheimer's. It's also sometimes there with people who don't have Alzheimer's. So like, it's definitely like it's always there in people that have Alzheimer's. So it's doing something, but is it causing Alzheimer's or is it a symptom of like neurons dying from some other thing? Unclear. This is like 50 years of research. Like people still haven't decided. But that's why there's no drug for Alzheimer's. Even though if like the plaques were the problem, we can get rid of the plaques. Doesn't always stop Alzheimer's, or like reverse the cognitive deficits of the memory impairment and stuff. So So that's the one thing with neuroscience compared to a lot of other diseases and other like fields. We know so little still, despite like hundreds of years of research. We often don't know what causes brain diseases of various kinds. Whereas like heart disease, you can probably like usually be like your vessels were clogged, like that is what caused the heart attack. Whereas like with like Alzheimer's, it's like we can't pin down like this is the cause. If we fix that, everything else is better. So all that to say that if you can target the disease, if you understand some aspect of how the disease came to be. Alternatively, you can target a symptom like clogged arteries or poor insulin or something if you like know that's part of the problem. And then finally, what is very rare to do in industry and like biotech companies is to target the biology sort of generally, which is much more of an academic way of doing it, where you're kind of like going after the fundamental biology of what's happening in that organ, as opposed to a disease-specific, symptom-specific.
So you just go back to like anatomy class then and just be like, oh, these two things connect. Let's see what happens when we poke it.
Well, sort of. So it's like, you can I think of it as like the easiest way I can think about it is if you have a disease, one way you could try to develop drugs is model the disease in the lab and throw things on it to reverse the problem. So if you like make plaques form on some cells in a dish, you could try to throw drugs on it to get rid of the plaques. That's one way you could test, right? So that's a disease model because you're trying to model what happened in the disease and then reverse or prevent it. And that's like the most common thing people do. So a lot of people, when they study cancer, they put cancer in a dish and they throw things waiting for it to die. What's going to kill the cancer is like the earliest way to discover something.
And then they're like, this bleach sure does wonders, but I don't know if humans can ingest it.
Right? That's where the drug development part comes in, where you like optimize it to make it like safe and like, actually efficient. Because, and if you think about it, like chemotherapy for cancer is basically like poison. Like it's bleach for your cells.
Yeah.
But because it's, your cancers generally are really energetic, they like die faster than your rest of your body. So chemotherapy and like radiation is basically just like killing things as much as possible and hoping you kill the cancer first. Things are better now. We have other medications, but
that's interesting that's like what's happening with the cancer, that it grows, like I'm assuming because it grows so fast, it's growing faster than the other parts of your body.
Yep.
And it dies faster. That's kind of cool. I mean, I don't know cool, but that's interesting.
Yep. And so that's the premise. And so that's like, I mean, chemotherapy and radiation are like a well-established treatment for cancer. But nowadays you can get like immunotherapies, if you've heard of, which is another kind of drug, right? It's antibody-based. So it's a biologic. So it's repurposing your immune system to fight off the cancer in a way. So it's called immunotherapy. Yeah. So the idea there is like, now we know some stuff about cancers, certain cancers, and we can make your immune system realize that's a cancer and use antibodies to bring your own immune system to the bad cells and get your own body to fight it off way more efficiently, or use that target to bring chemotherapy drugs to only the cancer cells and kill them off, as opposed to like radiating your whole body and being like,
I feel like that's just like biohacking right there.
Right? So that's the kind of cool thing where you like, as you like learn, like, oh, we learned how cancer cells evade the immune system to become cancerous, and then we can like hijack it to be like, no, like.
I'm just seeing in my head some weird info animation I saw once with like the shapes of the white blood cells, and it's like they don't recognize the cancer as an enemy, so they can't fight it off. And so that's the level of understanding I'm coming with. That's what I'm telling you.
I mean, that's more than mine. It's more than mine.
So for the cancer immunotherapies, it's kind of the T cells don't see it. They're kind of walking away being like, nothing to see here. But then now we can give like antibodies, which are basically little like bookmarks, to be like little flag to be like, no, this is actually bad. And so it's like,
that's crazy,
waving a flag at the T cell to bring it back.
We're talking to our body in this like strange language. It's very fascinating.
It's great. And that's what's so cool about biology generally, is that like your body's doing all this crazy stuff and we're just trying to figure out how it's working so we can talk and tell it when it's not working ideally, right? To like,
so you're like reverse engineering the disease way. You're like, okay, great. You're trying to figure out the relationship between the disease. Now we're just trying to figure out what happens to the body when we do stuff with it.
Yeah, or like you could think about it as like theoretically, if you can improve the fundamental unit of the system, you could apply it to any disease, right? Like if you could, like, so in our context of neuroscience, the 100 plus years of research have really identified a lot of the main proteins and how cells work in the brain. So we know a lot at like a very small, at a very focused level of how the cells are working. But it's so complicated, it's hard to pin down what exactly broke in the disease. So that disease modeling and trying to target the disease is very complicated. So our approach right now is forget the disease for now. We'll get back to the disease. Let's start with just what is that basic like function of your neurons to make functioning synapses. And so there's like a whole bunch of things involved in making a functioning synapse, like thousands of things are part of that. So we're trying to see if any drug candidate we're coming up with can make that better. With idea then that if you can make that fundamental unit better, you could apply it to almost any disease that has a negative impact on that fundamental unit.
And is this research going to lead to the Hollywood blockbuster Matt Damon movie Limitless, where we can access all of our brain? And
oh my gosh. That movie was actually good, but also like wrong, because like you are accessing all of your brain. Like every movie that starts with
Lucy
you only
also.
use 30% of your brain, I'm like, false. No,
just
like
at a time.
10%
lies.
No, it's like 10% of your brain is like neurons, but then there's like other cell types that are like fundamental to functioning brain that people like forget about. And I'm like, you use all of your brain. If you are not using some of your brain, you've had a stroke and you need some medical help. Like,
I'm pretty sure I have not been using all of my brain for a long time, and I haven't had a stroke, but that's other factors playing into it. Okay, I gotta ask one thing.
Yeah.
You said you make the synapses, so that means that you didn't like get a human synapses, right?
No. So that's the other really cool part about the work I'm doing is we're able to model human cells, human neurons in a petri dish, without harming any people or animals. So the idea here that we're using,
Who do you harm then?
We harm no one.
Just using anodes and cathodes and transferring.
No, so we're still using cells. So this is like they are human and mouse cells. So I guess some animals were harmed because to get brain cells from mice, there's only one way to do that. But for the human cells, no humans were harmed because we're using stem cell derived neurons. So the idea here is, and you guys did talk about this half correctly, I believe.
Sounds about right.
Where stem cells have been around for a long time, and everybody has stem cells. So this is a whole other tangent. I'm like, I can go on and on about stem cells because they're fascinating. But basically, stem cells, like I think you guys accurately said, are what give rise to everything in your body. So that's what allows you to have different organs. Like we all come from a single cell. How does that single cell become 200 plus tissue types? It's because the stem cell has the potential to become anything, and it grows really well.
That's why it's called a stem.
Stem. It is the stem, and it is the root of all the other
we got
cell
roots
types.
And stems this episode. I'm loving it.
It's very
much easier than I thought it was going to be, actually.
Yeah, and no, it makes sense. So the idea being that like, and I think Rebecca mentioned there used to be a big controversy around stem cells because originally the only place we knew they were there was in the embryo. So like you had to destroy an embryo from like maybe an IVF clinic or something to get human stem cells. However, we then learned that actually most tissues in your body have adult stem cells. So like, so basically there's like the root stem cell that can become like anything. And then there's like tissue specific stem cells that like, how else do you get skin that keeps replenishing? Like there has to be some skin stem cell that can like make more skin. So it can't make, heart cells, but it can make more skin. So then this discovery of like these like tissue specific stem cells let us understand more about stem cell biology and like the factors, like the communication that happens to like turn that single stem cell into like these like mature stem cells, and then the final
stem cell,
mature cell in the body that like does the final function. So with all that knowledge, we figured out a way to reverse engineer it. So now you can take skin cells, turn them into stem cells, like the the original stem cell, like the earliest stem cell, then tell that to become anything else in the body because it has unlimited potential. It's immortal. It can just live forever, divide into anything. It's immortal. That is the hallmark. All stem cells are immortal. Yeah. So then you can. So we take out some skin cells. There's a Nobel Prize for this. This Yamanaka factors turned like skin cells into stem cells and turned the stem cells into like heart, brain, liver, whatever. Like whatever your heart desires, you can make. If you know how it developed normally, you know what the steps were. You can put those steps on top of the stem cell and make it go through those steps and become, in our case, neurons. So we can have human neurons growing in a dish that came from somebody's like skin biopsy.
Crazy.
Yeah. So we have these neurons, and then they have functioning synapses, and then we like take our different candidates that we either like, come up with based off previous research or from like the literature. So a lot of important research happens in like academia and like different universities and research institutions. They kind of figure out a lot of those fundamental processes and are like, this protein does a lot of cool things in the brain. And we were like, does it? And then we like try it on our things and be like, it did cool things to the brain. Yeah. Like, and then that's drug discovery. And then drug development would be like, let's make it better and let's make it safer because, if someone's going to
drug discovery
take a
and
pill,
drug development are different things then. They're not I would have thought that they were the same world, but you're saying there's some people that are like, hey, let's figure out like what actually triggers something. And then the other people, totally different group tries to like narrow that down into the specific drug that they wanted it. That's wild.
Yeah. And it doesn't have to be different people. Like theoretically, if the drug I'm working on right now looks really good, I could start working in drug development. Because, but it's a totally different, like purpose. Because when I'm doing drug discovery, I have like a very simple system. I have my neurons, and I'm throwing shit on them to be like, here are certain outputs I'm looking to improve, like better synapses, more synapses, etc. Maybe happier cells, whatever. Like I'm looking for certain things. But that's a very simplified system. Eventually we want to get it into people, right? And at that point it's going to, and maybe before that into like mouse models. So that means it's going to be reacting with a lot of different things. And actually one of the biggest problems is safety, right? Like just because a drug does one positive thing, doesn't mean it doesn't do like a dozen horrible things. So like in a brain cell, it might make better neurons, but if you like put it in the body generally, it might cause cancer. So you have to be really careful about what biology you're triggering to induce that.
And even after all that, those commercials still have those speed reads at the end that it may cause nausea, heartburn, upset stomach, diarrhea, and death.
Right. So those are the ones that like were acceptable side effects.
And death.
Yeah.
But I think that's I put together the side effect thing, but yeah, I didn't put together that like when you're in the drug discovery phase that you also have to figure out those side effects, right? Because why spend all that time developing a drug if you know from phase 1 that, or whatever, from the very beginning that you can't give this to a human ever because it's always going to like shut down their liver or whatever it may be.
Right, exactly. So like some candidates we've worked on, like some of them are very specific to the brain, but like some of them would be like, yeah, it's making the neurons grow better, but it would also make other things grow better and grow more, and like you probably end up causing cancer. So it'd be like, ooh, it's having a really cool effect. And then my boss is like, yeah, but we're not going to follow up on that because the effort it would take to like separate out its like positive effects versus side effects would be like not worth it. Because probably the way it works is through that process, but like, you're trying to like, you can't really limit unless you're willing to take like brain injections every day. How do you limit which cells get which drug? Right?
As someone who's had brain surgery, it's a hard pass. Yeah, it's a hard pass to do that every day. Like it's not a good time. And I had minimal surgery, right? Like, and so I think, yeah, that's it's a lot to think about because I think when you first start approaching this, like as a layman, when you first start approaching this problem, you're like, oh, just figure out what works, right? Like do whatever you have to do. Like, why aren't the companies doing enough? Why don't they just have the answer yet? And it's like, well, yeah, we figured out the answer 500 times already, but actually none of those are feasible. So like, it doesn't matter how, like, it's a very specific combination of what is successful. not just what is right.
Exactly. Yeah. So I think I think like 90 something % of drugs fail before they get to clinic, before they don't pass, because clinical trials are like safety, efficacy, which is how well they work, and then like the final, like it actually does what it's supposed to do. So like you got to pass safety before you get anywhere else. And most drugs fail well before then. But if they, even like the most promising drugs often fail at the safety, because
safety first.
Because people are complicated.
So when you say you're throwing shit on the neurons,
yeah.
like, do you have like a petri dish with your neurons and then you're literally just dropping stuff on it?
Yeah. Pretty much. So so we're not really working on any biologics, which are like antibodies. So that's like one kind of approach, which we might do. Right now we're working with small molecules, like compounds. So like what you think of like drugs. So like aspirin, right, would be like a drug, a small molecule. And so we could make or buy different, these are like proteins basically. And a lot of them we select based off the literature. So people have already published that like protein X, when fed to mice for 7 weeks, makes mice smarter. We're like, so we can like buy or make that protein. And then we grow our neurons, in these like, technically 96 like dishes, but yeah, just think of really, 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 really tiny petri dishes. So we can do a lot of this. And then we would like the cells have like a growth media to keep them happy, because how else are they like? There's no blood vessels, so how are they getting fed? They're just sitting in their own little bath of
food,
piece of cheese,
of like of like liquid. Imagine like a really, really weird smoothie. Very thin. Nice. But like, in the body, your cells are getting nutrients from like your blood vessels, right? You're like oxygen and like energy is coming in from like the mitochondria.
I'm gonna kink shame. If you want to lay in a smoothie full of weird goop and just osmosis take in your food, like I'm not gonna kink shame. You're like,
yeah, okay, maybe not a smoothie, maybe like a soup. Think of it as a soup. It's very thin. It's very clear, but it is pink usually. Pink soup, bathing your cells, keeping them happy. So you need a specific temperature and specific nutrients to keep the cells happy and make them, like in our case, neurons. And so they're sitting in their little bath of soup. And
you like have a ladle?
They're neurons. So no, we have pipettes. We're pipetting. It's all. These are.
What's a? Oh yeah. Okay. Yeah.
I'm gonna my firstborn child.
It's basically. Don't do that. That's so mean. But these pipettes are basically like ways to move very small liquids volumes around, right? So we're adding like, teeny tiny, like little bits of liquid.
Get a teeny tiny ladle though?
A scoopula?
If I am coming to visit the lab, I request there be a teeny tiny.
That's for solids, though.
Yeah, that's what we have we have a little scoopula for measuring out compounds, like so we can like scoop the dry powder and make it into a solution. And that's where we do drugs like every day, but on the cells. And so so the cells are sitting in this like little soup of nutrients. And then we can add in whatever drug we think we want to investigate, and then we can we basically then measure the function of the neurons, and we can also look at the synapses and like quantify like the number or like the size, and based on like so many different like parameters, we can be like, yes, like drug A made things better, drug B did nothing, drug C killed everything, that's one out, like.
That was what I was curious about. Like, are you actually just physically looking at under a microscope and looking at the nature of it, or is there a way you measure its structure as a molecule? Like,
no, that's great. Those are great questions. A little hard to explain simply. It is a microscope for the most part. So I'm trying to keep it simple because it's like you can go down this whole rabbit hole. But yes, so on one hand, you can like. And you just really use a really, really powerful microscope and look at the synapses themselves. Like you can basically like label them so that like they turn a color. So that you could be like, because we know what's in the synapses. So there's like,
Can you paint with them?
Let's say 10. Can you paint
Can
with
you
them?
like paint with them if they're different colors?
You can label multiple different proteins with different colors, and then you could like, so there's like, so in synapses, like one neuron is the presynapse and one neuron is the postsynapse. So that's how you know they've started communicating, is that they have different proteins, but they're in the same location because they're like physically in contact. So you can label one red and one green and look for spots of yellow to be like, those have synapsed together because they are co-localized. So you can sort of paint with that.
A few times. I know what you're talking about.
Yeah.
I would
So much
totally
fun.
paint with them. It's like literally Pocahontas.
But you got to remember these are on neurons. So like you have the neuron and then the cell body, and then off the neuron are these like different processes called like dendrites and axons, and the synapses are like little like beads on a string, kind of dotting the processes. So, and it's the processes that kind of like go throughout your brain. So even if you have like one neuron over here, it might have a synapse way over here. And I mean, that's how your whole body works, right? You can have neurons in your foot that have axons that travel up your whole body into your spinal cord and then connect the spinal cord to your brain, right? They like, the reason you can feel your toes is because the muscles there have neurons in them that are connected all the way to your brain.
And do sometimes they like pass other places? Because like, you touch your ear a weird way and then you like feel it in your toe or something?
Yeah. I mean, people have been mapping out like where your neurons go, and most of them go basically from your muscles or from your like skin, for example, towards your spinal cord, right? So that's your peripheral nervous system, and then your brain is your center, and the spinal cord is your central nervous system, which basically stays where it is. But like, yeah. So just because you feel a tingling in like one finger doesn't mean it could be something somewhere in your arm. Like, it doesn't have to be in that finger, right? Like, depending on what's triggering the tingling. I don't know. So.
Right.
That's a whole complicated
I took you down a rabbit hole. I didn't mean to. Yes, we were looking at it under a microscope.
No, it's cool.
And
so microscopes, you can like look at how many synapses or how many neurons are synapsing to be like, well, the more the merrier, right? The more the synapse, that's generally means your brain is more connected and more is improved. So that's sort of like looking at the structure, right? That's the structural changes of the neurons of the brain. The other way is to look at function. So this is a little complicated, but basically calcium changes are a big way your neurons respond to signals. So like when you like pick up a pen and you like feel it in your fingers, the neurons are communicating to each other and passing up to your brain, and that's passing back, right? But that passing is like calcium waves going up and down to like trigger all these changes and trigger this effect. So basically, people figured out a way to label a version of calcium green. So when the neurons are active, they light up green. You can like modify the neurons to do that. So now in our microscope, we can kind of look at it at a relatively like high level, just like look at like thousands of neurons and be like, who's pulsing green? Like, are they pulsing together? Are they like not pulsing at all? Like, and you can think that like, and I wish I could show you a movie of this.
Jackie,
basically, we have movies where it'll be like,
yeah.
Right, where we'll have like early neurons when they're not fully synapsed, when they haven't made their synapses yet, will sort of twinkle by themselves, kind of like little stars. They're just sort of twinkling, but green stars. And then when they're more mature and they've synapsed together, like hundreds of neurons all just like pulsing together, yeah. The number of times early on when we got it to work the first time in our hands, like every time it would do that, I would be the backing like, because it would like. It'd be pulsing, and like one way to, you could, as an example, to measure that, you could, or like to use that as a way to decide if a drug is doing something. Our early studies, we would use known drugs to like prove our neurons are behaving like neurons. So like we could put in something like canic acid, which induces seizures, and like cells would be like pulse, and then we throw on canic acid, and they go like pulse, and they like freak the hell out because they're just so overstimulated, and we're like, yeah, they're working. Like that's the correct biology. That's how they should be responding to canic acid. And so that's an example. There are many, many, many kinds, but that's one example of the functional measurement of the neurons. So we can look for drugs that make them fire better or behave, like improve in their behavior over time, as a way to like say, this drug is doing something good. But yeah, if you ever want to Google like GFP labeled neurons, it's like cool.
So then the work that you're doing, it's, I guess, because it's so large, that's sort of just like, you're just going to keep finding more information and finding more information, finding more information. It's not so much like, oh, we have this one deadline or one type of drug or like a sort of end in sight to research.
Yeah, I mean, the end in sight would be like money. We always need money to keep going. Always helps. And like biotech research is very expensive. Some of the machinery is expensive, but it's more like the plastic consumables. We like burn through a lot of like plastic waste real fast.
You need to start using paper petri dishes, okay?
It's considered a biohazard. No, yeah, the plastic waste does make me cringe a little bit. But yeah, it's interesting question. We don't have a deadline per se in terms of like we're trying to get, like we'll reach a point and then we're like done. Like to be done, we'd have to like get the drug to people. Like that would be done, which is not really the deadline. So like the deadline, I guess, would be like we have a couple different molecules we're testing, different drugs we're trying to develop. We're trying to like see if this is a real. Like we've been working on this for like 2 years. We're like, we think this is a good option. Is it? So we're working on that. And then theoretically, if that works, we would switch into drug development and like improve on it, right? And so a lot of companies you hear nowadays will do, I think you guys even mentioned this in part one, where like we'll sell it off, right? Well, they'll like get a drug that works, that seems promising in like early discovery stage. And then other big companies like AbbVie or like Pfizer or somebody will come in and be like, yeah, that seems legit. And they'll like buy it from you or like license it from you. So that's one option. I think my company seems really invested in like doing a lot of it ourselves. So like we probably won't sell off any drugs we find interesting. We might like partner with people so that we have the money coming in so that we can do the like, 7 to 10 years of work it'll take to like get the drug to clinic. But yeah, there's not like like in a sense, we'll never stop working on it because there's always going to be something more to learn about it and ways to make it better and ways to like, if you find a drug that works, great. You can turn that drug into 10 more drugs, right? Like you will just want to keep.
And these are all to the aim of just
expanding
the neurons. I'm like, I can't even get to the next word. Neurons in the brain generally, not just like, so it could be Alzheimer's, could be any number of things.
Right. So that would be the next step, right? Like if, so like basically you can think right now we have like 3 molecules we're sort of working on. Like we're a company of like 5 people. We're like super small. But for example, like if one molecule like confirms to be like doing the effect we think it's doing, we could try to develop that and make it so that it's helpful for like dementia or like based on what it's doing to the neurons, we can be like, oh, it's making more neurons grow or like make them grow better. Well, that's like loss of those like synapses are a symptom of like of like dementia or like so that would mean like, oh, we probably want to target that. But if we find that, oh, this is actually really good at making like a different measurement, like maybe just strengthening synapses, so they're not making more, they're just like stronger. Well, that's probably something associated with like Alzheimer's or like, or if this is preventing cell death, like that's something you can associate with like stroke, like depending on what the final like effect we like are seeing with the molecule, we can apply it to different diseases for our best chance of getting it.
It's like a baby. It's like you don't know what it'll be yet. It has to grow up and tell you.
Right. Exactly. We don't want to like put too much pressure on it. We're going to let it find its way and let the data inform us on like what we can do with it. So yeah, actually it was funny. One of the molecules I'm working on I'm working on it because my bosses at a previous company were already working on it. So this is kind of going back to the idea of like repurposing. So they had found this molecule and they don't know how it works. They have no idea what it's doing, but it is really good at minimizing pain and it seems to give like better cognition.
We like those things.
And so like,
yeah,
those are great things. So I'm working on trying to figure out how is it doing that? Like, how is it making like in mice, making mice and people smarter? It's already in clinical trial. Like people are like showing improvements in memory. It is not opium. So one of my projects is to try to figure out how that drug works. Because if we figure out how it works, I could then make new drugs that target that same process. I can't use their drug because their drug already exists and is got like patents and stuff around it, but I can make a different version of it that targets the same
types of problem,
like process in the
you
body.
just walk around like you're in the Matrix, like numbers revealing themselves to you, and you could just like you pick up a yogurt and you're like, know what's inside of it. Like,
no, mostly it's me reading a lot of papers, being like, like 15 different people saw this one protein made mouse synapses 10% better. I wonder why, and like
so it's
trying to figure
more
that out.
reading than necessarily in the lab doing the throwing stuff on things.
I think I'm about like it changes because it'll be like I'll be throwing things in the lab and then be like, oh, I had a problem. I don't know what's going on. So then I go back to the computer and I go reading for days to be like, has anyone else encountered this problem? Do people see something similar? And then take what I learned, talk to my coworkers, go back to the lab, freak out for days. And so like right now I'm in the throwing phase right now in one of my experiments where I'm just like in the lab, like being like spinning this, like looking for color changes there, like looking for stuff. And then like, 2 months ago I was in like trying to read papers and I'm like, I don't know what you're doing, but I need to copy this to see if I can do it. And if it's real in my context, because that's another big thing people do, right? Is like, you'll be like, something, people discovered something in like, I don't know, rat neurons that do one weird thing. And you're like, do human neurons do it in the same way? Like sometimes. Yeah, so it's a mix of things. But yeah, I'm in the lab. I got my lab coat. I'm got the gloves on.
I'm glad you have a lab coat. That's important. As a doctor, you should definitely have a lab coat.
I think there's like a study that showed that people will like just trust people with lab coats.
100%.
Like even if they're not actually doctors or anything.
Blindly. Don't even ask a question. Absolutely. Do my gynecological exam. That's fine.
Oh no.
Wow.
I was thinking more like it gives also people confidence that like the moment you wear a lab coat, you automatically just feel like I know stuff. Like I do. I'm like I don't. But,
but hey, if the coat gives you that extra pep,
as you demonstrated, you know a lot of stuff.
Well, this is like 10 years of research, so I should hope I know a little bit.
Is there anything that we've missed that we need to, or something that we covered in the first part that we need to know?
I feel like we covered most things.
I'm a little scared to have to like do the recap.
I know she's stretching. She doesn't want to have to get. I
think we like talked about a lot of things that at least stuck out in my mind. I think one thing this is just interesting, not necessarily like a thing you guys said incorrectly or anything, but the bit about like Viagra and Botox. I just want to be like, those are really great examples, and I wanted to throw in like another one just to like maybe blow your mind a little bit, because my mind was blown when I learned this. So like you guys said, like Botox is a poison. Viagra was developed for like dilating like blood vessels in the eye to like improve like vision, and then side effects.
Dilated some other veins.
Exactly. So that's people like repurposing things based off of like the side effects for like medical use. And so one thing that was kind of cool I learned not too long ago was how people are now trying to use venom from like snakes as medical therapeutics.
Snake oil.
but like real like like so the idea being that and then I think there's maybe like 7 or 8 drugs currently in the market that were developed based off of like venom, and I think the example I remember, I think it was something like it causes it's from snakes and it prevents blood clots because the snake wants to eat the animal it's bitten, and people repurpose that as a blood thinner because it prevented blood clots.
Maybe I'm
so like venom. You might be on a type of repurposed snake venom. And so anyway, so I thought that was kind of cool. So that's one of the like quintessential examples of like repurposing from nature to help people.
I love that, because I've actually heard that the reason we say snake oil salesman is not for the original salespeople who were like ancient Chinese traditional herbalists, but that their snake oils might have had efficacy, but that it was the like colonialists appropriating it and not doing any sort of like knowledgeable tincture creation. But that is straight from Google's lips to mine, so don't take any weight in that.
Sounds real to me.
That's
But
fascinating.
yeah, no.
Okay, Jackie.
Okay, well, I have one more.
Okay,
I also
yeah.
heard that there's a lot of drugs that have come from like weird stuff in the Amazon too, like weird toxic and poisonous stuff, and that's where we get like so many of our drugs is the Amazon. I don't know if that's accurate either, but
I don't know how accurate to say specifically the Amazon, but there are lots of medicines that come from plants. So actually, kind of going back to like a lot of like ancient Asian cultures have a strong history of like herbal remedies, and like retroactively people learned that a lot of those were based off like real. It wasn't just like magic and smoke or whatever. It was like there are like I don't know if you've actually a friend was just telling me about this, how she learned willow bark.
That's
like
aspirin,
willow bark,
right?
as aspirin. It's basically the formulation of aspirin. It comes from the willow bark. So like a lot of like in India, there's like Ayurvedic medicine, and a lot of that, if you like look into the plants that go into those tinctures or whatever, a lot of those are the real medications today. Like
Wow.
it's the same. It's obviously like more mixed and maybe not as potent and yada yada, but it's like the premise is like scientifically sound. So identifying useful compounds.
I was going to say, I
mean, great. Don't do that.
We can appreciate both. We can appreciate the holistic and the researched.
Yes. So it's like if medications are like causing harm to your liver, supplementing them with some validated herbal remedies might help reduce some some of the side effects. But yeah, it's a very complicated mix of like oral tradition and like things that have not been validated versus like I can think of like a dozen things. Please don't. Oh my God.
And listen,
essential oils are not
Reddit does not count as peer review.
No, it does not.
Okay, I kid. Okay, are we gonna do this? Are we ready for this?
Let's get our safety gear on and hit the labs.
Okay, you start this one off.
Okay, so drug development. We're gonna.
Okay. It starts with 3 different things that you could start with researching: disease, symptom, and the target or the biology, aka.
Beautiful. Yes.
I choose biology.
Me too.
Okay. Disease. Oh God. T cells are like nothing to see here.
Oh, that was about cancer. That was about how cancer disguises itself in the immune system, and we've developed immunotherapies to recode our immune responses.
And you gave the antibodies red flags, and then they were like, "Yeah, I see them now." And then the T cells were like, "There are things to see here." See, my note was helpful. Thank you.
That was gorgeous. But then I'm still a little like, I feel like I would need a finer clarity to like really make a distinction between if you're developing something based on the like disease versus trying, as you know, we flipped things around and we're trying to target the biology.
I got this.
And those overlap. They're not like hard silos. Those go together. Yeah.
But I got this. Brain diseases are hard to understand because we don't really know what's happening, and so that's why the biology route for neurosciences is a good path to take over the disease or symptom, because stuff has plaque, and sometimes that's bad, and sometimes it doesn't mean anything. And if we just went out with if we just went after the plaque, it wouldn't be good enough. And so we get some human cells that are immortal, and they were at some point someone's lung, and we were like, be a baby cell, and now it's a stem cell, and now it's a brain cell, and that's the circle of life.
Wow, she is a genius. Nobel Prize when? Okay. Yes, because we're learning the structure of a biology and how to affect parts of it, and then how we can apply that to various treatment plans.
And we
then
test
it gets
it
passed.
with the rave. We test it. If there's a rave,
Yes,
then
we did
it's
the
doing
rave
good.
test. Yes,
And
we checked
if
our
it's raving,
synapses, we color coded them.
Yes.
and there was
exactly.
calcium changes, and we watched those calcium changes to see if things were actually happening. And then
Exactly.
all our GFP labeled neurons are synapsing.
Synapsing so hard that we pass it on into drug discovery, where then we can narrow down
No,
our
we're in drug discovery. We're passing it
compounds.
to drug development.
There you go. Yeah.
Okay. Yes. We have to
You
discover
discover it,
before
and
we
then you
develop.
develop.
That does make sense.
No, we can do whatever way we want because we passed
You
the
could.
rave test. So
You did. I feel that was an amazing summary. I think the only thing I was like, I don't think you described stem cells correctly, but everything else spot on.
I said
that
they
was
were
great.
immortal.
You did, and then you call them babies from lungs, circle of life.
They were little lung
And I have to
babies.
be like, no. Oh God, no. But yeah, immortal cells give rise to everything. There you go.
And they raised the roof at the rave, and that's what
Semi-permeable
goes down.
membrane. Okay, that's all I remember from my biology class.
I love that. That's what you remember. That's great.
Have we increased our score?
I would like to retake the course.
Are we PhDs? Yeah. Next we will when we edit this. Don't worry. We'll get a second shot on understanding.
I feel like you guys are a solid 8 now. 8 out of
Wow.
10. You went from like a 6 to an 8. Thank you guys.
I'll take that. I will take that. That's not a bad score.
Solid B+.
Thank you.
I love it.
Yeah.
That's one pill I can swallow. Okay, there I said it.
I will make more. You can swallow more pills.
She's so good at the puns every time. And sometimes like I don't even catch them. And then in editing I do catch them. And I'm like, oh, I didn't even laugh. I didn't even see it.
No.
Oh.
Gotta get a laugh track to prompt you next time.
Yeah, I need
Please.
that. I have been attacked by cats this entire call. So this is, but thank you so much for joining us. This has been, I think this is up there with meteorology on things that I just like, my mind has expanded, and then the amount that we have talked, I'm just like, wow, that's a lot of information that I never would have had otherwise. It's super interesting to know how that works.
Yeah, thank you, Dr. Devi. And hopefully maybe someone listening at home is like, oh, this is right up my alley. I love this. I'm so inspired and invigorated by science. I'm going to go right out and improve the world through drug discovery and development.
Join me.
Yeah.
But yes, thank you for putting up with I feel like I went to a lot of different things because I get excited and we jump topics a lot. So I threw a lot of information at you guys. And I don't know how well I explained it, but
I think you did
hopefully I have conveyed that the science is cool.
I love to peek behind the curtain. That's what it felt like. It wasn't like I was understanding it all, but it's things like we're aware of and that just so interesting and like important, it's just incrementally making the human experience a little bit better.
Yeah, like seriously.
That is the goal.
Like for I know actually tons of people who have been affected. I mean, obviously I've been affected by neurological disorders. I have family and friends with, dementia and Alzheimer's and all of that. And so like, I am so grateful to people who can understand the science, because like there's a minute that you were talking there and I was like, and this is why it took you so long to get through all the schooling, right? Because like the things that you were explaining, I was like, how you even know that is just bonkers, because there's so many layers and you're like, I won't even go into it. And I was like, I can barely handle what you just told us. So like, thank you for making it through all of that schooling and like having that knowledge and saving the world.
Oh my gosh, trying to one brain cell at a time, I guess.
Yeah, are there any things you want to plug? Any things people can do to find out more about your company or promote it? Anything like that?
Nothing to promote about the company. It's still in stealth mode, but hopefully in the next year or so we'll go live. But yeah, I hope people were engaged and enjoying to hear about all this random neuroscience drug discovery stuff. And I think getting involved in local science outreach efforts is one of the most exciting ways to learn science without actually going to school for 10 years and having to struggle through a PhD. So I'm involved in a couple of different mentorship activities, and I highly recommend people find local events in their communities to hear their neighbor scientists talk about their different kinds of work. So
That's super cool.
check out your local schools.
Very cool.
I'm definitely going to. And you said you were going to give me a tour of the lab sometime, so I will give a little short update some point on what that's like. Bex, if you're ever around, we'll get you in on this action, and we'll get those lab coats on, and people will trust us.
For sure. You guys can pose next to the microscope. Be like,
Bex and I are doing Instagram posts. All right. Devi, thank you so much. We appreciate you. Let's go be scientists.
Doctor, that was great. And that was the show. Thank you everyone for watching, and we hope you stay curious.
If you enjoyed this video, watch our other videos here on YouTube, or you should listen to the episode on We're Experts Now podcast. Bex, where can they find us?
You can find us wherever you listen to your favorite podcasts like Apple Podcasts, Spotify, and iHeart Music.
And what if they want to talk to us?
If you have ideas for something you want us to investigate, send us an email at we'reexpertspod@gmail.com. That's wereexpertspod@gmail.com.
Great job on that spelling. A+. Do you do this professionally?
I believe we're both doing it professionally right now.
Wow. People should know about this.
I completely agree, and you can find us on Instagram at We're Experts Now Pod.
Thanks again, and if you enjoy the show, subscribe and comment, and do not forget to leave a 5-star tip.
That's a great idea. Are you a podcast expert?
I think we both are. I think
we're experts now.
This has been a Just Being Better media production.